Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Stuart Gibb, PhD, Scientific Strategy Lead from Terumo Blood and Cell Technologies.
Thank you very much, Alex. It's a pleasure to talk to you this morning. I'm going to be talking to you about the cell expansion benefits of using hollow fibers. So first off, I'd like you to brief you with an overview of our technology. If you look over on the left-hand side here, you'll see our Quantum Flex platform. If you've heard of the Quantum platform previously, Terumo has had it on the market for about 10 years now. And Quantum Flex is the latest updated version of the platform, and we launched this at the end of September. As you look to the middle of the slide here, you'll see the key element of what we're going to talk about today, which is our bioreactors. We have two bioreactors that are available for the Quantum Flex platform. We have a standard bioreactor, which has been on the market for about 10 years now. And for Flex, we have a new bioreactor. The standard bioreactor has a surface area of about 2.1 square meters, and the small bioreactor is approximately a tenth of that. Above the bioreactor, you see how the bioreactor disposable set looks like out of the box. You can see the bioreactor at the bottom there. In the middle, there is a tubing organizer, and there are also attached bags at the top. The Terumo Quantum platform is functionally closed, so how we add material on and off of the system is by sterile welding. As you look over to the right-hand side, you'll see the door of the Quantum opens, and the disposable set fits into the middle there. And as you can see, the bioreactor is in the middle of that section. In order to explain to you how our hollow fiber biotechnology works then. So as you recall from the previous slide, the standard bioreactor, the larger size there with the 2.1 square meters. Each one of those bioreactors contains approximately 11,500 hollow fibers. As you can see on the right-hand side of the screen there, the hollow fibers are kind of arranged and stacked into those bioreactors. The diameter of each hollow fiber is approximately 200 microns, which is quite a large lumen size if you think of an average car key cell being in the range of 8 to 12 microns. So the hollow fibers are actually semi-permeable. They have a molecular weight cutoff for about 17 kilodaltons, which allows you to extravasate either into or out of those fibers. Items below 17 kilodaltons have the propensity to pass across, so you think about small solutes such as glucose or any waste products we can direct either into or out of those fibers. Within the hollow fibers, the space is known as the intracapillary space, and outside of the hollow fibers, the space is known as the extracapillary space. And I'll explain more about that in the next few slides. So digging more into the concept of how our hollow fiber bioreactors work, and put together a couple of diagrams here to kind of walk you through. Again, thinking about that standard size bioreactor compartment illustrated here in box 1. So within those fibers, that's where the cells live. That is the cellular compartment colored here in gold. Again, that's our IC intracapillary circuit. Outside of the hollow fibers, where the cells do not live, is a blue circuit in these diagrams, and that is also known as the extracapillary circuit. So moving on to box 3, as you can see, HFBR is the abbreviation for the hollow fiber bioreactor. And what you can tell from this slide is that both the IC and the EC circuits pass through the hollow fiber. At the bottom of the blue circuit there, you'll see this item labeled as GTM, which is the gas transfer module. So again, the system is functionally closed, and in order to oxygenate the media to supply your cells, we take care of that on the EC circuit. So one of the benefits of hollow fibers, our bioreactors, is that we have flexible feeding options. So you can either use one media source to supply both circuits, or you can use 2 different media sources. One media source, for example, if you use suspension cells, so you supplement that with cytokines, you can have that only on the intracapillary circuit, that was where your cells live, whereas the media that you would use for the EC circuit passing through the gas transfer module, you could use a base media. So the access to fresh media is a very powerful option as you consider how to handle certain elements of your cell culture. Feeding can occur just on the IC circuit alone if you want to use that one type of media, or you can use the EC circuit if you want to limit the circulation through your IC circuit, and you can feed via the EC circuit if you wanted to limit the access to the speed of the media, say. Feeding can be constant or periodic. For example, if you use adherent cells and you want to have no flow through the IC circuit, you can pause the pumps so there is no flow, and you can just feed via the EC circuit. For suspension cells, for example, the T cells, we can change the flow through those circuits through the IC circuit, and we can employ a bidirectional circulation, keeping the cells within the hollow fiber bioreactors. As you can see, there is with those circuits, media would pass out of the hollow fibers and then return from one side and return to the other side. However, if we employ a bidirectional flow, that keeps the cells within the bioreactor platform. Circulation can be low in the IC circuit in order to prevent harming cells, but you can utilize the EC circuit to maintain oxygenation, feeding, and waste removal. As alluded to, you can use those 2 different types of media, right? So your media with cytokines, you would use on the IC circuit where your cell lives, and a base media you could employ on the EC circuit. And then if you look on the bottom right there, you'll be able to see that you would use a minimum amount of media containing cytokines, whereas the rest of the media would just be your base media. So how we maintain that critical microenvironment for your cells then. So again, if you kind of limit the flow through the IC circuit, you can feed, you can augment feeding from the EC side. So you can see in the feeding slide here, we've got glucose passing into the lumen where your cells live. If you have expensive cytokines for 17 kilodaltons or above, they won't pass out of the hollow fibers. So you can see the 17 kilodalton at the top of that slide there. But if you have cytokines that are below 17 kilodaltons, you may wish to put them on both the IC feeding circuit and the EC side also. For waste removal, removal will pass down the concentration gradients. So items such as lactate will pass out of those hollow fibers into the EC circuit. And then you have options of how you handle the waste there. So you can see in the diagram on the left-hand side, there is a straight blue line to the waste bag because we tell the computer to open the valve to the waste bag, and that passes through the out of the EC circuit, whereas that dotted line on the IC side indicates that that waste valve is not open, because your cells are there, and obviously for suspension cells, we don't want them going to the waste bag. So you have options there of how you drive media into the system and how you pass media out of the system. So key features for suspension cells then is that you do have that option of using 2 media formulations, and if expensive cytokines do not pass through the hollow fibers, remember that molecular weight cutoff piece. Once maximal feeding rate is reached in the IC circuit, you can employ the EC circuit to supplement low molecular weight substances such as glucose. And that bidirectional flow piece I aforementioned, that will keep cells within the hollow fiber bioreactor. In addition, waste substances from the IC circuit will then going to diffuse across into the EC circuit and will be taken out into the waste. And the waste valve for that particular scenario would only be open on the EC circuit. So just to summarize those principles then, so Terumo's blend and cell technology functionally encodes hollow fiber bioreactors are comprised of semi-permeable hollow fibers. These hollow fibers have a diameter of approximately 200 microns and have a molecular weight cutoff of approximately 17 kilodaltons. So that value it's not a cliff value. So if it's 16, it will not necessarily pass straight across there. And even at 17, it's going to depend somewhat on the flow that you pass through the IC circuit. So if you have a low flow and it's around about 15 or 17 kilodaltons, you probably lose less than 5%, for example. And with those 2 circuits, the IC and the EC circuit, you have flexible options for feeding and waste removal in order to maintain critical parameters for your cell type. So now we've gone through some of the principles of how hollow fiber bioreactors work. I'd like to give you a working example of how those cells look on the platform. So in this example, we're going to show you some data on T cell expansion using the standard bioreactor, that 11,500 hollow fibers with the 2.1 square meters. And throughout the data, I'm going to show you some analysis of the phenotype that we've generated. To explain the paradigm for you then, so from a PBMC set of cells, the CD3 positive T cells were immunomagnetically isolated. They were then cryopreserved until the time we wanted to run the quantum system. Prior to loading onto the bioreactor, they were activated with soluble T cell activator. And then we used 3 different donors for this set of data. The expansion on the quantum bioreactor platform took between 7 and 8 days. Obviously, there's donor-donor variation, hence the change in numbers there. And again, with those flexible feeding options on the IC media, we used media with human serum and IL-2, whereas on the EC side, we just used that base media. Again, 2 different media choices. At the back end of a 7-day expansion, the harvest generated between 13.1 and 24.7 billion CD3 positive T cells, which represents about 8.1 to 9.7 doublings. And obviously, key important to this these expansions is the viability, and we saw over 96% viable cells at the end of those expansions. As we all know, for those who are going to focus on T cells, phenotype is important. Phenotypes of T cells range from being naive to stem cell memory T cells, central memory cells, effector memory cells, and terminal effector memory cells. Of course, as we think about CAR T, then the T cell stem cell memory subset is considered the T cell subset critical for CAR T therapies. So one of the important things that we wanted to look at off the bat was the stability of the subsets throughout the manufacturing process. And I'm going to walk you through a couple of pieces on this slide first. This is talking about the hollow fiber bioreactors in this particular study is just one aspect of what the study was trying to look at. So if we just cast our eye to the bottom of the slide first, you'll see in the bar charts that there's 4 panels. So in the first column, that is, we took samples from each of the different stages and did analysis on those samples. So you can see we have a pre-expansion, that's obviously pre-expansion, pre-loading onto quantum. Then there's a post-expansion column for when we when we harvested cells from the quantum. After those pieces were taken care of, we then place these cells into another device that we have called the Finia fill and finish system. Those cells were aliquoted into bags of cryopreservant, and then those cells were frozen. So you can see that in post-thaw, those cells were thawed out after those cryopreservation procedures, and samples were taken at that point for analysis. And the last column there, you see that samples were rested for 24 hours in culture, and a final piece of analysis was taken care of. So the red boxes there, that's going to just kind of illustrate and draw your eye to the fact that we are looking at the samples pre and post expansion on the hollow fiber bioreactor. So as you can see, this was a very enriched population of CD3 positive T cells. It's approximately 100% throughout there. And then looking at the subsets, you can see CD4 and CD8 subsets were stable throughout the expansion. Looking at the T cell memory generation of the cells as they went through the manufacturing process, if you look at the key on the top left there, you can see we were interested in analyzing the relative portions of naive T cells, the T cell central memory cells, and the T cell effector memory cells. So you can see, for just an example, if the cells are highly positive for CCR7 and negative for CD45R0, then they would be considered naive T cells in that particular case. And you can see these were the cells were gated on CD3 first, and then CD8 or CD4 thereafter. And you can see on the columns on the right-hand side that the proportion of CD8 and CD4 central memory cells increase throughout the manufacturing process. The C cells stem cell memory subsets are also kind of generated, right? So further gating strategy, again, splitting between CD4s and CD8s. So gating on CD3s, then CD8s, and then CD45R0. As you can see in that top kind of right-hand quadrant, that would be representative of T stem cell memory as a subset of T cells. As we go through the manufacturing process, as you can see, there's an increase in enrichment and representation of those cells throughout the expansion. Obviously, the field is very much interested in whether cells are terminally exhausted or not. An increasing appreciation of exhaustion for T cells tells you that if cells are positive for both PD1 and TIM3, then they would be considered to be towards the exhaustion end of production for those particular cells. As you can see in the columns on the right-hand side, there is a very slight increase in exhaustion markers, ranging between 1.7 and 11% for the different donors. But as they go into those cryopreservation pieces, what we talked about, and forward out, and you think about that in a clinical relevant setting, that there is minimal representation of a co-expression of those exhaustion markers. So the cells are kind of being enriched for the central memory and stem cell memory cells. They're not terminally exhausted, but are they functional? So how this set of data was generated then, so the samples were taken at those 4 different time points as previously discussed. They were then activated overnight with CD3, CD28 dynabeads. And then after 24 hours, supernatants were collected and analyzed for cytokine assays. As you can see, with the cytokines listed here, their IL-2 slightly increases throughout production on the bioreactor. But when you think about being ready for a clinical paradigm, and you can see 24 hours post-4, after it's gone through the entire process, IL-2 increases, and similarly with interferon gamma. And obviously for TNF alpha, there's a modest increase there, but also kind of going in the right direction that you would want. So then just to summarize, so Terumo Blood and Cell Technologies hollow fibers bioreactors have flexibility in the options for maintaining those critical quality attributes for both suspension cells and adherent cells. We have two bioreactors. We have a standard size bioreactor with a surface area of 2.1 square meters, and that was kind of used in the data that I presented here this morning. And then we also have a small bioreactor novel to the new Quantum Flex platform that we just launched, which has a smaller surface area of 0.2 meters squared. Just to give you a kind of rough idea of what it equates to, 2.1 square meters is equivalent to about 120 T175 flasks for those people distilling flasks, and the small bioreactor is approximately just over 11 T175 flasks. Both those bioreactors are amenable to the autologous cell therapy programs out there, and also if you think about additional experiments such as PD experiments and seed train programs. So the data of what I presented here shows the critical quality attributes of T cells are maintained or enhanced throughout expansion on our hollow fiber technology. And I'll pause there and I'll take any questions. And thank you very much for listening.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what is the difference in this solution compared to a Zuri or a stir tank with ATF?
Good question. I mean, really, it's a range of options that we have with the hollow fibers, right? You can, again, you can grow either adherent cells or suspension cells on our bioreactors. You have flexibility in the options of how quickly you exchange, media. You have different options for, whether you want low perfusion on where the cells are drying or no perfusion, right? You can feed via a separate circuit. So we do have a lot of flexibility in those options as you kind of have critical parameters in mind that you want to maintain. Yeah, a lot of options that we can employ in quantum.
And is the small bioreactor suitable for adherent and suspension cells? Excuse me.
Yeah, no, we've got preliminary data that is works fantastic for both adherent and suspension cells. So yeah, both the small bioreactor is again it's 1/10 of the standard bioreactor that most people have known about. It's been around for about 10 years. But the small bioreactor works on the same principles. It has that similar IC and EC circuits. You can employ the same kind of feeding strategies and kind of applications as you think about whether you want to do adherent suspension cells, transductions or differentiation. All those applications do translate down with the small bioreactor.
Our next question is, how do you get the cells out of the bioreactor?
So there is a harvest procedure that we employ. So everything is, we have either preset computer programs or you can design your own on Quantum. I didn't get into some of the kind of technical pieces of how you operate it from a computer point of view, but you have a program that harvests, which governs the harvesting of cells. So for suspension cells, it's quite simple, right? We just circulate around the loop and then open a valve, tell the computer to open a valve and puts that into a harvest bag. And then it's very quick downstream, it takes about 60 seconds. And then for adherent cells, you'll start out well the bag that contains like a dissociation reagent, That goes into the bioreactor, incubates for whatever time you want. We have recommendations on that, and then after which you circulate again fluid through there. Your cells are detached, and that places the cells into a harvest bag, and again you do your downstream processing. So it's all automated. The system is completely automated. You design the protocol, so again we can supply you with a preset program.
And how do you know that you're achieving high clinical efficacy with high cell density?
I mean, really, it comes down to those key parameters, right? We know the cells have got a high variability. We know that functional and downstream assays indicate that there's no impediment to growing cells on the quantum. So the cells have come off as being very, very much high quality cells.
Is the hollow fiber filter type fixed, or are other pore sizes available? Have any other cell types other than T cells been used in the system?
Oh, we have many examples of different cell types that are being used on the platform. The system really is agnostic to what cells you want to grow in there. We help you tailor your procedures, whether it's adherent for coating reagents or different types of suspension cells. We usually tell people that it's like, you're the experts in your cell type. Tell us what's critical. We're the experts on how to run the device, and we can meet in the middle. So we tailor protocols with respect to critical parameters that the customer is aware of.
And what factors in the bioreactor change when working with adherent cultures?
Is there a further explanation on that? I guess, I mean, as far as density goes, we have recommendations on the density of cells to seed to begin with. If when we load cells on the quantum, we don't have fluid passing through that cell circuit for the first 24 hours. We keep it very still. Let the cells attach to the hollow fibers. We have recommendations on coating reagents if people have particular coating, looking for recommendations or have their own coating reagents in mind. And then we just feed through the EC circuit during that attachment period. So it's very much different way of handling cells for adherent cells, but it's again, we're kind of well versed in whether it's a suspension or adherent protocols to recommend.
All right, well, it looks like we're running short on time, so if we don't get to your question, we'll be passing them along to Stuart, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what is the percent recovery of cells from the hollow fiber using your harvest procedure?
So we see I would say within 5% of the number of cells we expect to get off of the hollow fibers. One thing I didn't discuss was how we measure the amount of cells on there. We use lactate as a proxy, and so we have a mechanism of working out an estimated number of cells. Generally, what we see in the harvest is that we're in 5 to 10% of that number. And again, it's a calculation number, which we can't actually view the cells once they're on those hollow fibers. Again, it's a very compact space for a large amount of surface area.
All right, well, thank you, Stuart.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.